First of all, I did want to say thanks to the speakers and thanks to Randall for orchestrating the workshop. It's been incredible. High quality talks with such a serious scientific treatment of emulation. Now, as part of the tech review project, and wanted to cover a few things here today. I'll be talking about what I mean by the brain mapping problem, touching briefly on its major challenges, talking a little bit about brain mapping, current initiatives, a little bit about economic drivers, commercial opportunities, and then just to finish on a selfish note, I was not expecting the quality of the questions that we've had. I was a little starstruck by our speakers, so I'm looking forward to having a discussion to wrap up all of you. So, David famously described and whether the organization of realized in silicon chips in the population of China or in beer cans and ping pong balls does not matter, as long as the functional organization is determined. And
John, we're having trouble with the audio. Maybe we could take a pause and come back.
Sure.
Okay, let's go ahead and switch to Bill Crugen, who is in charge of our computational brain models section. Sorry to jump right to you, Bill. Can we maneuver things around there at the live stream or the live audience? So I'll do a brief introduction of Bill while he's getting ready. Bill is a Louisiana native who studied computer science at Carnegie Mellon University. And he is currently working toward his PhD at UC Berkeley for advanced studies. You're on, Bill.
Hey, can you hear me?
I can hear you, yeah.
Okay, great. Cool. The background, maybe if I move. All right, I'm gonna try to avoid some feedback real quick. Okay, let's try this. So we're talking about the section of the technical review about computational brain models. And basically the way that this fits into the bigger picture is you've heard a lot about neuromorphic hardware on which we might implement some of these things. You've heard a lot about how we scan a brain and try to get a good 3D reconstruction of the brain. But even if we had a perfect three-dimensional reconstruction and the world's greatest supercomputer, we would still need to know the equations and how to describe how neural activity changes going forward in order to actually run some type of emulation of a brain. So this section will just kind of get at how we derive these mathematical models and talk about some of them that are being researched today. So there's three main sections that we'll talk about in this section. The first and the main focus of this will be on single neuron models. So how do we just model the activity of one neuron if we know all the inputs and try to generate the outputs? And theoretically, if you can get a really good single neuron model, then you can just combine a bunch of them together for to model an entire circuit of neurons. And then I'll also talk a little bit about system models. So if we try to abstract away from the activity of individual neurons, can we still model how that system behaves over time? And then finally, I'll briefly touch on some of the current large-scale simulations that are in progress and and what they contribute to the field. So on the left here, we have maybe the most simple single neuron model that many of y'all might be familiar with. This is what you'd see in artificial neural network. And it's inspired by a neuron because it has inputs which come from other neurons. It does some transformation on them and it has an output. This is a very successful neural model in things like deep learning nowadays. It's used in those artificial neural networks. And it's so successful because it's extremely computationally efficient. So we can implement this on a GPU and run hundreds of thousands of these, millions of these in parallel, and it works really, really fast. But it exists on the spectrum of trying to balance computational efficiency with biological realism. And obviously for something like whole brain emulation, we need both computational efficiency and biological realism. And this model that I've introduced on the left doesn't really capture a lot of the biological realism of a single neuron. And just to illustrate what it's missing, here on the right are two example neurons from two different parts of the brain. And you can see the top one has very different morphology than the bottom one. And presumably these two neurons have evolved these morphologies because they perform different computations in the brain. The shape of these is presumably very important to the computations they perform, and you couldn't just take the top neuron and put it in place of the bottom one and expect that circuit to work the same. So all of that variation between different neurons is kind of lost at the cost of computational efficiency in these artificial neural networks. So we're talking about, in this section, going into where current research is on trying to balance these two things and get the best of both worlds in terms of modeling biological realism, but also retaining that efficiency. And I'll briefly just touch on a couple of examples from recent years. So on the left, if you were on this live stream earlier, you saw Ted Burgess talk about his hippocampal prosthesis. And this is from one of those related papers talking about how to model individual neurons' output in terms of their inputs. And here, he's abstracted away from every little compartment of the dendrites of different neurons by just considering each neuron in the input as one point with a certain spike train, so a certain pattern of inputs, and then modeling just a very simple output spike train of that neuron. And so this can be done very efficiently just by implementing these functions directly on a CPU and not worrying about maybe modeling the entire complexity of the shape of different neurons, but the math is such that you can still capture a lot of the computations that are happening within that single neuron. And so that strikes a good balance between biological realism, computational efficiency, by doing that abstraction of one layer. And on the right is just another example, getting at the same question a little bit differently. So this lab is trying to basically model what different types of computations happen in these dendrites. And here, this is from their paper where they looked at different types of inputs on different dendrites and whether those inputs were spread across the cell and how correlated they were in time, and basically different variations of inputs that you would see in a brain. And they tried to extract some computational rule of how the neuron was exactly integrating these inputs and constructing an output. And so this again gets at, we abstract away from neurons and come up with something that retains as much behavior as possible, while allowing us to efficiently implement it. So we can balance those two competing drives as much as we can with single neuron models. But there's another possible answer to this dichotomy, which is to what if we can abstract even away from individual neurons to try to describe the behavior of a whole section of the brain, a whole neural circuit at once. And so on top is kind of one example of this type of thinking. So this is a deep convolutional neural network, and these were originally developed for, or I should say, gained a lot of success originally by doing visual input processing. So you show it a picture, and the picture input is on the left here, and that picture is processed more and more by these different layers of artificial neurons as you go from left to right. And the first layer might respond to something like very low-level features like a line in the photo. The second layer will respond to slightly more complex features maybe like a circle, and the third layer will respond to more complex features, until eventually you go down this entire list of layers and you get to the end. You have maybe one unit that responds to if the entire picture is of a dog, and another one that responds to if the entire picture is of a cat. And so this is a very successful architecture, and it's based on, originally was inspired by trying to model the visual system. So on the bottom is a very oversimplified version of the human visual system, but you have the same overall architecture where you have the input is an image that goes into the retina, and then as it goes through the thalamus and then into the cortex with these different areas, V1, V2, V4, you see a very similar architecture at first glance, where V1 neurons tend to pick out individual low-level features of images, and then V2 neurons will pick out slightly more complex features, V4 neurons slightly more complex features. And so the advantage of these system models is, you can imagine if this was all the visual system was doing, was ultimately going from visual input to very complex features and categorization, then we could implement that entire system much, much more efficiently with a system model like the deep convolutional neural network that we designed from the ground up to be computationally efficient, and it would recreate the same behavior. Obviously, in this particular example, the visual system is a little bit more complicated than this. It does a lot more things than just pick out whether there are pictures of a dog or a cat. And so that's why these deep convolutional neural networks don't describe the entire behavior of the visual system. But this just serves to illustrate why these system models are important to develop, and down the road, as they're being refined, how they might come into play in some type of emulation environment. And finally, I'll just briefly touch on this. So most of the section will be about the different models that people are developing, and then I'll also touch on the models that are currently being implemented in things like the Blue Brain Project and Allen Institute for Brain Science Human Brain Project. So these are kind of the closest things we have so far to validating these the behavior of these models as they're applied to very large neural networks on very big supercomputers over a long period of time. And so I'll touch on kind of the results that each of these organizations has contributed to our understanding of computational brain models. For instance, the Allen Institute for Brain Science implemented 2 different patches of cortex that they were modeling, one with a very simple point neuron model, which is equivalent basically to the on the first slide, that very simple artificial neuron that I presented, and then the other model they did a what they called a biophysical model, which is much more complex and takes a lot longer to simulate, but reproduces the activity of individual neurons much more with much higher fidelity. And so then they were able to compare and contrast the different behaviors that came out of these 2 models and talk about where some models were appropriate for some questions, but maybe failed at answering other questions, and maybe didn't reproduce the full behavior of what they were expecting. So that's that'll be the final thing we'll talk about it in this review section. And with that, I'm more than happy to answer any questions that you might have about anything I've discussed, or I'd love to hear what else you think would be interesting to talk about in this section.
Thanks, Bill. The online Q&A is quiet again, but Keith did post a question. He says, is Bill just describing a standard perceptron or some modern variant?
Yeah, basically just a standard perceptron. So I guess what he's referring to is this artificial neuron from the beginning. And yeah, I was just putting this as an example of what would be one quote neuron in one of these large neural networks as a very oversimplified version of a biological neuron. But yeah, I think in most of these cases it is implemented as a perceptron. Right, but it totally doesn't capture kind of the grander complexity of these individual neurons.
Other than that, it's still pretty quiet online. Do we have any in-house questions? Other than that, it's still pretty quiet online. Do we have any in-house questions?
So, when you're not talking, Hi, sorry, Alicia, did you ask about in-house questions?
Yeah.
Yeah. So I don't know if there are in-house questions by others here, but I certainly have a few questions of my own.
Alicia, did you ask about in-house questions?
Okay, keep that from doing this twice. So I'll just start and ask a question. You know, you went through a number of different models. Thank you, Bill, by the way. Thank you very much. And it's terrible that we're not doing the clapping for the people who are doing this tech review. We probably should. So Bill, you mentioned these different levels at which you can do a model, different types of models. And I was just wondering, because single neuron model seems like an easy thing. Like just do a single neuron model, and then if you can put the individual neurons everywhere, great, the whole system should work. But is that actually true? I mean, has anyone ever made a good single neuron model that really predicts the activity of a real neuron?
Well, I guess a lot of that answer depends on what you qualify as a good prediction. So it depends how you want to validate that model, right? If you, for instance, you can do something like Ted Berger's hippocampal prosthesis, where your model is validated by being able to recreate in an actual brain with your model of a neuron, some activity of a real brain. So that's one level of validation where, yeah, you can make a single neuron model that does kind of function in some way. But the question of whether it functions perfectly like a real biological neuron, I think, is very difficult to get at, because there's so many levels of complexity, right? I don't think we've even totally captured as a field how dendrites fully contribute to how a neuron operates. For instance, there's some, for a long time, the field kind of considered dendrites to be these passive input receiving areas of the neuron, and then everything gets sent to the body of the cell, and then computation happens. But now there's a lot of research coming out about active dendrites, where they're actually actively performing computations in the dendrites, and I touched on that a little bit. And of course, there's even subcellular things like the exact organization of receptors on the cell, and different transcription regulators that occur over the course of neural activity. That is crazily complex to model, and I don't think we even have a good idea of how to validate one of those models yet. So the answer, I guess, would be on some level, yeah, we've implemented these single neuron models, but definitely not to the level of what you might consider like perfect biological realism.
Don't forget to mute yourself.
Thank you. Sorry, Diana, did you have a question? Is it? Oh, I think we still have time. Yeah. So you want to come ask it? You can ask it straight into the microphone. Yeah.
Hi, can you hear me? Thanks very much for your talk. Okay. So I had a question about, it's sort of a follow-up question on the previous one. I was just wondering, in terms of system identification for single neurons, what do you think is the correct way to start? You mentioned all these details that probably play a role, like ion channel distribution, neurotransmitters, active dendrites, and so on. So let's say, I mean, there must be, in principle, there should be an equation that can describe the behavior of a single neuron. And how would you go about adding parameters? Is there a certain order to it, like in terms of importance? What contributes to that? And especially when we add functionality and behavior to that. Yeah. Thanks.
I think a lot of it is probably limited by what we actually have experimental access to. So we're only very recently being able to probe things like the exact arrangement of receptors at a synapse with very fine detail, very fine detail resolution, and actually being able to see that in real time and correlate kind of the structure to the function, as people discussed earlier. That's something that we're still definitely working on. So in terms of what you should model, I think currently the cop-out answer is it's what you can model, is right. If we have enough data to basically validate whatever your predictions are, then yeah, you should include that in your model if you're looking for perfect biological realism. In terms of kind of mapping out what would be the most kind of important features of a neuron to model, I think that's not totally clear right now, because there's, as I mentioned, there's a lot of different interacting things, and even things like the how DNA is regulated in response to neural activity. I think is a hugely potentially complex interaction that hasn't been explored in nearly as much detail as many other aspects of neurons. So I think it might be too early to tell exactly where we should go, but luckily we don't directly have to answer that question necessarily, because it's kind of just whatever we can access. We need to develop tools to get better temporal resolution, better spatial resolution on these things. As soon as we can get the experimental methods down to investigate quantitatively how these neurons act, then yeah, we should include those observations in our model as well.
I've got another question from Keith on the online Q&A. If richer bio-inspired or morphology representative models still produce the same spike train behavior, then doesn't that just suggest we don't need those richer models? Or are there further theories of neural behavior that would require such models even if they fire the same way?
I suppose if it perfectly recreated the relevant output, then yeah, you're absolutely right. You wouldn't need the morphological details. I guess it depends on a couple things. What do you actually consider your relevant output? So maybe the exact spike trains for a short amount of time that you validate on are correct, but you still need to capture things like plasticity and how the spike trains will develop over time, and maybe that's not captured in one of the simpler models. It also depends, right, how you're going to apply these models. So for things like the hippocampal prosthesis, again, that we heard about earlier, it seems that just recreating the estimated spike trains does recover a lot of the function. So if that's kind of your metric, then yeah, that's as deep as you need to go. But if you have other metrics, such as trying to maybe simulate things for drug discovery or something on a lower level, or things like the neuromuscular junctions, where a nerve attaches to a muscle, if there's some kind of organization there based on the exact orientation of the receptors and things like that, that would obviously require maybe a lower level explanation. So I guess it's difficult to answer exactly where we should stop in terms of complexity of modeling. That's kind of one of the big questions of mind uploading and whole brain emulation. But yeah, totally depending on what your application is. If you consider your relevant output to be, say, spike trains, and you have a model that gives you good enough spike trains to whatever measure you want, then yeah, that's the simpler the better, and then the more computationally efficient the better. So that's totally application dependent.
Great. So things are still pretty quiet online. Do we have any more in-house questions for Bill? Randall, if you're talking, you might be muted.
Okay, so I was muted apparently. I was just saying I had a similar question, and I think Brian already did a good job of answering most of that anyway, because I was going to ask something along the lines of how can you evaluate the promise of a given model, how to test its possible use in brain emulation. And I think this kind of comes down to the same thing. You know, if you can only record from a certain proportion of neurons, then, for example, it's really important that you can try to model to some degree what you don't know, that you can model the rest as some kind of noise or some sort of other contribution that you can't actively record. And if your output is producing still a spike train that is plausible, that follows the same kind of probability distribution, then you're fine. Or maybe not. And again, this depends a lot on what we learn as we go along. So I think Brian already answered most of that by just saying, we have to dig in and find out why some things are more useful or less useful in a given case. Do we have any further questions on this in the house? Anyone there? Nope. Okay, so we have no more in-house questions on this topic, Alicia.
All right, we have no more online either, so maybe now would be a good time to switch to the new live stream link. All right, where can I tell people to go for this new link? We'll go to John Doe's talk after the switch. Everyone will want to go to carboncopies.com/live, carboncopies.org/livestream for the new link. Randall, if you're talking, you're muted.
Thank you. People should head over to that link, to carboncopies.com/livestream. That's sorry, carboncopies.org/livestream. I'm just reading it straight off the chat and making the same mistake Alicia made. So go back to the original page on the live stream page. You find a new URL there. That's the one that we're going to be using from here on out, because this one is expiring. And I guess we as speakers here are going to receive a new URL for the Hangouts as well, I assume. Okay, right, we're making the switch now.